Okay, we're ready to go. Okay, so today we're going to talk about everything you need to know, everything you need to consider before designing constructs for molecular biology. So we're going to focus on design, all these things you need to think about. A lot of this is going to be review. I'm going to go into more detail on some other things. And hopefully, if you have a question, you can just ask. Okay, so the first thing you need to consider before you design anything, right, is the fundamental purpose, what you're trying to do with your molecular biology construct. Hello. So, right, like there's all kinds of things you want to do. There's deletions, which we talked about. There's insertions, which is like adding something. There's transforming a plasmid. We can make changes. So like we can make mutations. We can insert tags. You need to know exactly what your purpose is, okay? That sounds obvious, but think it through, okay? So now let's talk about mechanisms of insertions. Mechanisms of insertion. Okay? So, a lot of ways that we insert foreign DNA, transposons, into new organisms in biotechnology is with transposons. What are transposons? Never heard of them yet? It's okay if you haven't. This is good then. Nobody told you about like the Indian corn story? Not even in like the food science?
Oh no.
Seriously?
Yeah.
Okay, then I gotta go into that rabbit hole. There's a woman named Dr. I'm gonna spell her name wrong, but her name is Barbara McClintock, something like that. And she won a Nobel Prize for discovering transposons. So transposons are what we call jumping genes. So in corn, if you think of corn and you think of Indian corn, okay? You guys have seen Indian corn, right?
Yeah.
Okay. So in Indian corn, the kernels are different colors, okay? How is that possible? Well, there's genes that regulate color. And there's also jumping genes in that genome. And the jumping genes will sometimes jump into the reading frame of the color genes, breaking them. And now certain colors shut off. Does that make sense? So Dr. McClintock won the Nobel Prize for discovering that these jumping genes exist. And the way that she studied this was by looking at different colored kernels in corn. I'm sure it's more complicated than that, but that's the basic gist. So let me talk about exactly what these are then, since you guys haven't heard of them. So there's a gene, a single gene, and the name of the gene is either called transposon, and a transposon encodes for a protein, which is an enzyme. Enzymes do what?
They're catalysts.
Sometimes they're catalysts. Yeah, they're catalysts of reactions, right? It's a perfect way to say it. And the protein that this codes for is called transposase. Okay, so when you see A's in biology, that means it's an enzyme, like phosphatase, kinase, transposase. That's an enzyme. You're right, it's a catalyst. Okay, and what does this protein, so let's say this is a transposase protein, okay, some globular structure. What does the enzyme do? So the transposase has sequences. I'm just going to draw it funky like this. And these sequences can recognize DNA. And oftentimes it's at a very specific site, what we would call a consensus sequence. Do we know what that means? That means like it recognizes a certain specific sequence of base pairs. Like, let's say the consensus sequence was ATGC, ATGC. That means it recognizes that. Does that make sense? That's what it binds to. Now, that's just an example. I'm not saying it recognizes that specifically. Now, when you see transposons, what you see is you see these recognition sites flanking the gene on the 5' and 3' ends. And this transposon ORF makes a protein that grabs its own gene and cuts it out. So the transposase, the verb, the catalyst, it's a nuclease, and it's cutting this sequence out of the DNA, and then it moves it and inserts it. Inserts it somewhere else in the genome. So let me show you what would happen. So let's say you got corn, Indian corn, and here is chromosome, we'll say 1. This is a transposase in red, and here are the recognition sites. Transposase gets transcribed into messenger RNA and translated into a protein, which grabs its gene at the recognition consensus sites and pops it out, and it can bring it to a new location and make cuts in that DNA and now insert it into that chromosome. So it jumped from here to here. Does that make sense?
Why would an organism do that, though? Mess with its own genes, maybe shut something off.
Very good question. Goes back to the selfish gene theory. The transposon is not part of the organism. Okay, it's not a gene that helps the organism. So you're right. It's actually a gene that hurts the organism. All the transposon cares about is itself. And oftentimes these transposons can make copies. So oftentimes they can copy paste. Okay? So this is what we call parasitic DNA. Okay? And if a gene can replicate itself, it's going to, because that's what it wants. Now, the organism, just like you said, the organism doesn't necessarily want that, because if it's jumping into constitutive housekeeping genes, what's going to happen to the organism? It'll die. Exactly. So there's a selective pressure on the organism to streamline, to get rid of these selfish elements and oftentimes break them. So oftentimes these things will get mutated. So if you look at your own genome, the human genome, you can find thousands of these broken transposons from old infections, and they got there and they infected your DNA and then they just died there. Okay? So that's a very, very good question. And the answer is that they're two separate things that are competing with each other. The transposon cares about itself, and it doesn't necessarily care about the organism. It just wants to replicate its own DNA. And sometimes that is at the cost of the organism. Okay, so now that you know that these transposons can grab DNA and move it, now you can imagine how we can hack this for biotechnology purposes, and we can use this for our own purpose. So if we make a plasmid, transposons work by having two plasmids. Plasmid 1, this is biotechnology transposons. So when we use transposons in biotechnology, we are hacking the transposase and changing its cargo. So in a normal wild-type transposase, it's grabbing its own gene, okay? But if we understand the biology of the enzyme, we can change its cargo. So let me show you how we do that. And we do that by utilizing two plasmids. One plasmid is called a helper, or another name for it is a suicide plasmid. And I'll explain that. The second plasmid is called the donor. Okay. So on the helper plasmid, we put a promoter that turns on. And in the multi-cloning site, we put the transposase. Okay? But the recognition sites that we talked about, we remove those. Okay? And we put those recognition sites on their own plasmid, a different plasmid, the donor plasmid. Okay? And then in the donor plasmid, we put gene X. Now what's going to happen? Now we inject or get both these plasmids into a cell. So we add this and we add this. What's going to happen?
Instead of cutting plasmid 1, the transposase cut plasmid 2.
Exactly. So transposase is on, so it'll get made, it'll get transcribed into messenger RNA, and it'll get translated into protein. But instead of the protein grabbing the transposase gene, the protein is going to grab gene X. And it's going to pop gene X out of the donor plasmid, and it's going to insert gene X onto a chromosome. Does that make sense? So this is an insertion mechanism. We can use this. We can use transposons to insert a gene of interest onto a chromosome. Now, the reason plasmid 1 is called a suicide plasmid is because nothing of this plasmid survives in the organism. This plasmid is just injected just to transiently make the transposase to help the insertion of gene X. And in gene X, none of this plasmid survives either, except the gene X that gets cut out and inserted. Does that make sense? So these plasmids are made to die. They're a delivery mechanism of delivering gene X. So we might as well talk more and more about transposons and talk about some of the considerations of transposons. So one of the good sides of transposons is if you find one that works, it works pretty well. And you can find ones that work in bacteria, and you can find ones that work in eukaryotes. Okay? So they work pretty well. But let's talk about some of the downsides. So one of the downsides is that the insertion, the location, so where it's putting the gene in the genome is random. Okay? We can't control that with the transposon. So you will, it will sometimes randomly insert itself right in between an important gene, and that will mess up your experiment. Okay? So the downside is that it's random insertion. We don't know where it's going. Okay? What's the other downside? The other downside is that what works in bacteria does not always translate to eukaryotes. And what works in, say, insect 1 or plant 1 might not work in insect 2 or plant 2. So they're not always readily translatable from system to system to system. Okay? So those are some of the downsides, and those are some of the positive things about transposons. Okay, so that's one mechanism of insertion, is one transposons. Let's talk about a second mechanism of insertion, which we've already talked about a little bit, which is recombination. Okay. So let me talk about this. Let's see. First, we'll talk about this in the context of yeast, and we'll review. So recombination requires homology. And just like a transposon, it's a way to insert gene X into chromosome. Okay? Now let's compare this directly to the transposon method. Is recombination site-specific?
Yes.
Yes. It's regulated. We can control, so it's site-specific. That means we control where it goes. And how do we control where it goes?
Choosing specific homologs.
Right. Choosing homology regions. Okay? So, I'm just thinking of how bad. Let's talk about two examples of recombination. One we'll talk about in Drosophila, and one we'll talk about in yeast. Okay, so let's do Drosophila first. So in Drosophila, when we nowadays make transgenic Drosophila, when we want to insert gene X into the Drosophila genome, we use a site-specific integrase, which is a form of recombination. So what's an integrase?
Enzyme.
It's an enzyme because it's A's. What would you think it does?
Integrates genes into the genome.
Yes, exactly. It integrates genes into the genome. Where would you find a wild integrase? What organisms want to integrate their DNA into a genome? Viruses. Exactly. So what we've done in Drosophila is we've taken a virus integrase. So viruses often want to make their own genomes and they want to integrate those into the host genome. So we've taken a virus integrase, which is called Phi-C31 integrase. Good morning. And Phi-C31 integrase recognizes and matches specific homology regions, and they have names, ATTB and ATTP, okay? And Phi-C31 integrase specifically catalyzes recombination between these two sites, ATTB and ATTP. So let me show you this interesting system that a guy named Cohen-Venken has set up in Drosophila, which we all now utilize. So what Cohen-Venken did is he made a system first starting with a transposon. Okay? And he made a helper and a donor. The helper, just like I said, has a promoter on that makes the transposase. And here are the recognition sites in the donor. And in the donor, he put ATTB, the site that Phi-C31 integrases, recognizes, okay? And then he, step one, micro-injected these plasmids into Drosophila eggs. Now tell me what's going to happen.
It'll get randomly put on their genes.
What will randomly get put into
The ATTB.
the ATTB? So what he did is let's say this is fly one. ATTB gets inserted right there. Okay? But he did this 1,000 times. So he made 1,000 of these flies. Fly 2, fly 3, fly N. And all of these flies had random insertion points for the ATTB. Okay. So now he's got 1,000, I'm approximating, whatever, 1,000, maybe he had 300, maybe he had 10, whatever. But now there's a lot. So originally he made a bunch of these flies that have sites where ATTB is inserted. Okay. Now step two. Now, before we always had to do Drosophila transgenesis with transposons. Now what we do is let's say we take fly one. Now instead we make a plasmid that has ATTP. Remember, ATTP matches ATTB. And we put our gene X on this plasmid. And let's call this plasmid P-U-A-S-P-A-T-T-P. I think I might have flipped them, B or P. It doesn't matter, though. What you need to know is just the concept. So now we put this gene X on this plasmid, which has an ATTP recognition site. And now what we inject is Phi C31 integrase protein plus this plasmid into Fly1. What's going to happen now?
It's going to have gene X at whatever site was ATTB the first time.
Yes. So this protein, Phi C31 integrase, is going to grab ATTP and it's going to match it with the ATTB site and it's going to induce a homologous recombination. And now what's going to get inserted is your plasmid with gene X. And now what we've done is we've built a system that is site specific. It's only going to integrate at what's called the landing site, which is the ATTB site. Does that make sense? So what Cohen-Venken did is he built 1,000 flies with a bunch of different landing pads for this integrase. And we can pick any fly we want with these different landing sites at different locations. And now we can insert any gene at that landing pad. Does that make sense? Yes.
So does that mean that the gene won't be passed on to every single member of the population? Because it's only on one side of the chromosome?
So here's what will happen. Let me see if I can answer this. So here's what will happen. What you would do is you would inject, you'd basically inject like 100 eggs. Okay. And the way that we select for this is going a little bit more advanced right now, but the way that we select for this is on this plasmid, there's actually a gene that changes the eye color red if this insertion happened. So then we would take, say, one fly that survived these injections that grew up with red eyes. Red eyes. And that means that in that fly, at least one of those chromosomes had that insert. Right? Okay. Now what's going to happen is if you pass this fly, it's going to follow Mendelian genetics. So you're going to mate this to a wild type, okay? And it's going to do like a Punnett square where you have insert, wild type chromosome, wild type chromosome, wild type chromosome, and you're only going to get 1/4 offspring that have the insert. So no, it's not going to be 100%, but here's how you make it 100%. That next generation, this is called the F1, that next generation, you kill all white eyes, kill all, I was going to make a joke. Kill all white-eyed flies. So this one would be red because it's wild type plus insert. Oh wait, it'd be 1/2. It'd be 1/2. This one will be red because it's wild type plus insert. These ones will be white. So you get 1/2 this first generation that will be red eye. Now, in the next generation, you just kill all the whites and you only let the red eyes reproduce. Okay. So then in the next generation, you do the Punnett square again. And now what you have is you're mating a red eye to a red eye. So you have a red, and let's assume it's heterozygous. So you have wild type, but now you have red, wild type. So now this one is going to be red, 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 white. Now it's 3/4 red, right? Does this make sense? Now you kill all the white eyes, you mate them again and again and again and again. You only take red eyes, and then eventually you convert the whole population to what's called homozygous red, which is your transgene. And now you don't have to worry about it anymore. Does that make sense? So that's how you get a unanimous population of a transgenic fruit fly line. But that's more that's more just trying to answer your question. What you really want to know is you want to know this mechanism of insertion, that there are these enzymes called integrases, and their function is to catalyze recombination events at specific sites. And they're found from viruses, and now we utilize those for site-specific recombination in biotechnology. Does that make sense? Okay. Second way of recombination, we'll talk about the yeast again. This is review now. Let's go over here. Okay. So let's just talk about the yeast recombination again. The yeast, and how is it different? Well, the yeast has its own enzymes that do recombination. Okay? And if you put in a plasmid, let's say we want to recombine, just like we talked about, we want to recombine gene X into the chromosome. What we do is we build a plasmid with gene X, and we put homology regions here that match sites on a chromosome. And then when we transform this plasmid in, we can select for recombination events where that plasmid recombines into the chromosome. And now we've inserted gene X. And this is utilizing the own enzymes from yeast. Yeast has its own enzymes that do this. You don't need to add 5C31 integrase. The 5C31, Drosophila has its own enzymes that do this too. We all have enzymes that do this. The reason we use the 5C31 in Drosophila is because it's site-specific. And we can designate it to land at these landing points. And it's very efficient. Okay? In yeast, we designate the site-specific spot by building homology regions. And how would you construct a homology region? I just want to make sure you guys understand that.
Each sequence its gene DNA and look for a place.
Yeah. So you'd have to have its sequence genome. That's correct. And you would look and you would pick a spot. And let's say you want to pick this spot. You would download the sequence, and you need about 30 base pairs, and you'd pick 30 base pairs, and you could literally add them onto your PCR primers. You could just build them into your PCR primers when you clone gene X. You put them right here. And then when you PCR that and clone it into the plasmid, now it's got those recognition sites on the 5' and 3' ends. And you can do an insertion. Okay? So the basic gist is that mechanisms of insertion. 1, transposons. 2, recombination. Let's talk about 3. Viruses. So bacteria is a good example for this. Here's a bacterial cell. In like 1950s, 60s, 70s, we were just learning how to add foreign DNA into organisms. And the first organism that we did this with was E. coli. And the first way that we did this was using a virus. So a virus is a thing that will inject its DNA. And there's a subset of viruses called bacteriophages. What does that mean? Yes, exactly. They target bacteria. They try to kill bacteria. So bacteriophage, or short, phage, is a virus that infects bacteria. And there's one specific one that we use and originally used in E. coli, which is called Lambda Phage. Lambda Phage is famous because it has 2 life cycles. It has one life cycle where it integrates and waits. So it just sits in the genome. And then it has a second life cycle where when things get bad, when shit hits the fan, it jumps out of the genome. It jumps out. And it makes particles. Okay? But most of the time, it just wants to sit. And see, the phage is content because if the phage can insert itself right here, if the phage can insert its genome into the bacteria, the phage gets its DNA replicated for free on accident by the bacteria. The bacteria, when it replicates this chromosome, I should draw a circular chromosome, it accidentally replicates the phage. So the phage gets a free ride if it can integrate into the genome. So that's what it prefers. So this lambda phage, we originally used to transform E. coli. And what we did is we could purify the phage genome from particles. So we could induce the bacteria to create particles. And then we would purify the phage genome, which is a little tiny, almost like a plasmid. And then we would put our gene X into the virus genome. And then we would infect those particles into bacteria. And that would integrate into the bacterial chromosome. And now we've integrated gene X. And we didn't really have to do anything except use the machinery that was already built into the phage. The phage has its own integrase. It has recombinases. It has its own, sometimes it has its own replication machinery. So one third way that we can insert things, mechanisms of insertion, is utilizing viruses. Okay? Still on the first line. It's okay. This is a fun discussion. Okay. Now let's talk about point 2. So those are the 3 things you might want when you're going to engineer a plant or an insect. How you're going to insert your DNA. Are you going to put in a plasmid? Okay, it's mechanism 4. Forgot this, mechanism 4. Sometimes you can just use a plasmid. A plasmid will work in a eukaryote like yeast and E. coli, but it's not going to be stable in something like a fly. Plasmid will not work. You would have to integrate into the genome. So that's the catch with plasmids. Okay. So those are mechanisms of insertion. Now let's talk about delivery. Okay. So what do I mean by this is, so we've made our constructs. We're going to insert gene X. How do you get it into the cell? Okay. So in a single cell organism like bacteria or yeast, budding yeast, I should draw it like this, you can do what we've talked about, which is called transformation. And we'll have more on that later. But this is just a means of getting the plasmids into the cell. Okay? And bacteria, we also learned, do conjugation, which is a way where they make these tubes, and sometimes they'll take up foreign DNA from the environment. Or sometimes they'll penetrate a neighboring cell, and they'll pass genetic information back and forth, which is called conjugation. Okay, so those are different mechanisms of delivery. But does this work in a plant or an insect? No. Like, what's one reason why? Can you think of a reason why, just transforming a plasmid into plant, why would that not work?
They're multicellular. So
yes.
One gets changed.
What's the only cell that we care about delivering the genetic material to? What's the only type of cell that we care about? The gametes. It's not going to work if you transform a leaf, because the next time that plant passes its genetic information to the next generation, you lose that transgene. It's not going to work if you transform the eye of an insect. You need to transform the gametes, because those are the only cells that pass on to the next generation. So that's a special process. That's not so easy to do. Which cells are the gametes of a plant? What are they called? They're called the seeds, right? We would have to deliver the genetic information to the seeds. Or, now this is a little bit, there are ways, when we talk about plant transformation, there are ways to micropropagate different things like that. So you don't necessarily need it to do the seeds for the plant. But for insects, or like a pig, if you want to make a transgenic pig, you can't cut off a pig's arm and grow a new pig. right? So you literally have to transform the sperm cells or the egg cells of the pig to get that thing to pass on to the next generation. So for insects, that means what do we need to collect? Gametes. We need to collect eggs. Like you're literally like, we need to collect eggs. And then what you can do is insect eggs have a shell, just like a chicken shell. And we do what's called, quote unquote, microinjection. And this is the same thing that they do for zebrafish and mice and pigs and sheep. Everything that is a multicellular organism that is an animal, you have to basically microinject eggs. Okay? So what you can do is you can make a needle, and in that needle, you are putting your plasmids. Let's say you're doing transposon delivery. Wake up!
How did that come?
Sorry, I could not resist.
No, I mean, yeah.
Okay. When you deliver, let's say you're going to do the transposon method, you would put two plasmids, plasmid 1, plasmid 2, where one would be the helper, one would be the donor, and you would microinject those, and then helper would make transposase, which would grab its gene and then pop it into the chromosome. So that's one mechanism of delivery, microinjection, or transformation if you're talking about individual cells. Okay, so let's talk about, when you talk about bacteria or yeast, there's chemical transformation, and there's electroporation. So what do you think chemical transformation involves?
If you denature like its cell membrane, right?
Yeah, good. So you're basically adding chemicals that, in theory, form pores in its membrane, and those holes allow foreign DNA to get into the cell. That's chemical transformation. So for yeast, that means you're adding lithium. And for E. coli, that usually means you're adding divalent ions, which is like calcium chloride or magnesium chloride or rubidium chloride, things like that. They in theory, cause these holes, and they can pick up DNA. And then sometimes in these processes, there's a heat shock, and you kind of like scare the shit out of the bacteria, and then it's like, oh my gosh, and it takes up DNA. It's part of the process. But that's chemical transformation. What's electroporation?
It's the same thing about electricity.
Yes, you shock them. So you take bacteria or yeast, and you put them in an electroporator, which when you press a button, shocks them. And the shock induces holes in the membrane, and then now your DNA, which is negative, starts like running and shoots in. Does that make sense? Now there's actually another way that we'll talk about more later, gene gun. Gene gun. What's the gene gun?
It's a gun that shoots genes.
Yes, it literally is. It shoots little beads with DNA bound to them. So you take little particles and you bind DNA to them, and then it literally like bombards the organism with particles and shoots the DNA into the cells. That's gene gun. You do that with plants. Okay, so those are mechanisms of delivery. I'm trying to think of what we go to. We talked about promoters. You guys understand constitutive inducible promoters, correct? No? Okay. What's a constitutive promoter?
Are these the promoters that are kind of always on? Like
Yes.
housekeeping?
Always on. Can you think of an example? Did you say RNA? No. Well, let's say you're saying the promoter for RNA polymerase. Is RNA polymerase always on? No, because the cell is not always transcribing messenger RNA. There's cases where it wants to shut that off. What is the cell always making? Well, one of these things is actin. This is a common one. Actin is a component of the cytoskeleton, and it's always on. So people will take the actin promoter, and they will clone the actin promoter in front of gene X, and then it's constitutively on. There's no regulation. It's just always on, full blast.
So you use that when you're like trying to use the cell to make a product or something.
Yeah, so here's a good example of this is our suicide plasmid, our helper. We'll put actin promoter in front of that transposase. And then as soon as we put that plasmid into the cell, it's making the transposase. Does that make sense? So that's a good case where we actually use a constitutive promoter. You know inducible promoters, right?
The tryptophan.
Yes, like the TRIP, the LAC, the ERA, operon, those regulators, those are inducible promoters. Weak promoters, strong promoters, that just means sometimes there's promoters that induce very, very low expression and sometimes very, very strong promoter expression. Actin is a very strong promoter. So these are things you need to think about is, do you have the right promoter for the right application? Let's see. Okay, let's talk about tags. Okay, what is an affinity tag? And why would you want to put one in a construct? What's affinity mean?
It likes something.
Likes something, yes. So when you study a protein, I've said this multiple times before, when you study a protein, you have to have a way to get it, to hold it in your hand, to use it, to purify it. Okay? And you do this by adding an affinity tag. The most famous example is the His6 tag, which I think I've talked about before. What's the His6 tag?
Binds to metals.
Yes, it binds nickel and cobalt. So what you can do is you can take, here's ATG of your gene X, and you can make an N-terminal His6 tag by inserting ATG, which is a start codon, a new start codon, cat, 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 cat. And this codes for methionine, histidine, 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 and then your protein starts. So then when your protein gets made in your bacteria, you can lyse the bacteria, which kills them, releasing your protein into the soup, supernatant, and then you can take that supernatant and you can run it over a column with nickel beads. And what's going to happen?
The protein you want will stick to the beads.
The protein you want will stick to the beads because of the His tag, and everything else will wash away. This is affinity chromatography. You've heard of that before in chemistry? Affinity chromatography. Now, how do you get the protein X off the beads? How do you get it off? This step is called elution. How do you do that?
Add something that has greater affinity to.
Yes! You add imidazole. You don't need to remember these well, you should, but you don't need, it won't be on the test. You add imidazole, which has a higher affinity for the nickel and the cobalt than the histidine tag. So then now your beads are bound to imidazole, and your protein X washes off, and you collect it in a little tube, and now you got a tube filled with protein X. Does that make sense? That's an affinity tag. There's also GST is another affinity tag. There's a whole bunch of these. GST is glutathione S transferase. And glutathione S transferase, what do you think it binds? Glutathione. So you can buy glutathione beads and you put a GST tag on your protein. There's a bunch of these different tags that have pluses and minuses. The his tag is quick and dirty. Quick, dirty, and cheap. Just like I like it. Okay. Secretion tags. So we will put, if you want protein X to get out of the cell, you can put a tag that will make it get secreted. This is going to depend on your organism's secretion system, SS. You'll see this a lot, SS. Type 4, type 1, type 2, type 3, type 4, type 6, SS, secretion systems. They all have these different tags you can use to get your protein out. Solubility tags. Let's talk about that. What's that? What do you think that is? What do you think is a very common issue in recombinant protein expression? You want to study protein X. What happens oftentimes with protein X?
It might be nature.
Yeah, it's insoluble. Sometimes some of these proteins, say you want to study a membrane protein. Membrane proteins are only soluble in membranes. So it's not going to be soluble in like a watery substance. So insoluble proteins precipitate. How do you study a protein that every time you try to make it, it just precipitates into a glob? Like that's not, you can't study that. So the way that you can study these things is you can add a solubility tag sometimes. So one of these tags is we often use is ubiquitin. What's ubiquitin? We've heard this before, I think. What's ubiquitin? It's a 7 kilodalton peptide modification. And ubiquitin is one of the most soluble proteins you can find. Okay? So if you have protein X or gene X that's insoluble, sometimes people will put the ubiquitin fused to the N or the C terminus. And now when you make that gene, when you make that protein, it no longer does not precipitate. Because the ubiquitin stays in solution. It like holds it in solution. Does that make sense? That's a solubility tag. Another one of these that you'll find is MBP, which is maltose binding protein. You'll see that a lot. It's a solubility tag. Sumo tag. Sumo tag is a ubiquitin variant. It's like ubiquitin, does the same thing. Okay? So those are solubility tags. 2 minutes left. Fluorescent tags. What would that do? We've talked about this before.
They glow.
They glow. So you can use this to study where a protein is. You fuse it to GFP. So you start ATG, then go into GFP, and then start your methionine of your protein X, as a fusion. And then wherever your protein X is, you'll glow green. Those are fluorescent tags. Caution with tags. Why?
They might mess up the function.
Yes. Anytime you add something new to your wild type protein, it's no longer wild type. Okay? So oftentimes when you do the experiments like this, you need to do controls. You need to do an experiment with GFP plus protein X, and you need to do the same experiment with protein X without GFP to make sure that the behavior is the same. Because when you start sticking things in front of your protein, you can mess up the function. Okay, we can finish here with codon optimization. And this should be review. What's codon optimization?
Certain organisms prefer certain codons.
Yes, certain organisms prefer certain codons. And why is that? What's the mechanism of that? Why would they have this preference? It all comes down to the concentration of what molecules?
tRNAs.
tRNAs. tRNAs are coded by genes, and each of these tRNA genes has a relative transcriptional level. And some organisms have lower transcription of tRNAs that they don't use very often. So if you code a protein with a bunch of rare codons that don't match, sometimes your protein will not get translated because the cell cytoplasm will literally not have the tRNAs to do that. I'll finish here by talking about an interesting plasmid. So there's special cells called Rosetta cells. And the catch, the reason you buy the Rosetta cells is they're bacteria, they come with a little plasmid called P rare. And P rare encodes all the rare tRNAs. So if you are worried about codon preference in E. coli, you can just use Rosetta cells, which make a bunch of tRNAs. All right, let's meet back at the lab.